So, dear students, welcome to our next lecture on a new chapter related to biotech only. This chapter is biotechnology and its application. So, in the previous chapter, we understood the recombinant DNA technology, okay, understood what all tools are used there, right? But in this chapter, we will see how those tools of recombinant DNA technology are actually applied to, make our day-to-day lives better. So, a short takeaway from what we learned from the previous chapter, we saw that biotechnology techniques means genetic engineering, recombinant technology. They're actually meant for making industrial means making products, biopharmaceuticals, then biologics. By biopharmaceuticals, I mean various kinds of drugs, right? Biologics like antibiotics, vaccine, etc. That those techniques will help us make these kind of products, and that too not in lab scale but in industrially large scale, means in thousands of liters. Okay, and it is all possible through the use of genetically modified microbes or fungi or plants or animals. So using genetically modified organisms, we can make biopharmaceuticals and biologics at a very large scale. Okay. Now, where are areas, in which areas biotechnology has made its mark? So in therapeutics, means development of new kinds of drugs for various kind of diseases. Biotech has played very important role. Then for diagnostics, means now we can detect various kind of cancers or other genetic disorders at a very early stage and start treatment, okay, and thus increase the chances of survival. Then crops are now being genetically modified so that they give more yield. Food is processed in such a way that the shelf life is increased as well as the nutritional values are enhanced. Then for bioremediation, biotech is playing a very important role for using microbes that can digest the toxic byproducts of petroleum and other crude oils, okay, and thus helping us rid of pollution, okay, of the nature, of the water and air and soil. We can actually reduce this pollution by using bioremediation techniques. And that all is coming from the biotechnology principles. Then also various kind of microbes and other organisms are used for treatment of waste, like the waste coming from your homes and industry scale. So those are converted into non-toxic byproducts and then only released in the environment. So biotech has made this possible. And lastly, for energy production, that to green energy production, biotech has really helped us. So for increasing food production, means we are now going to see how biotech has actually helped us in agriculture, in the field of agriculture. So the main goal of all farmers and all those who are concerned with, agriculture and food production, they want that more and more food should be produced so that the growing population of our country can be fed and people are not starved, right? So to reach this goal, biotech has really helped us. So we can actually increase food production, that is increase the use of crops by either using chemicals like pesticides, herbicides, fertilizers, etc. But it has a drawback. First, they are very expensive, which a small farmer cannot afford, and also these chemicals, they have harmful effects on the environment, means it can permanently pollute your groundwater, and ultimately this groundwater when reaches your homes, you drink it and you're ultimately affecting your health. So this kind of harmful effects can be is one of the, is due to the usage of these agrochemicals. Then what other options are there? There is organic farming. But again, organic farming, although it's a very sustainable option, it doesn't involve polluting the environment, but the yield of this technique is quite less. The amount of food produced of organic farming is comparatively less. And last option that we have for increasing food production is using genetically engineered crops. Okay. So among these three options, the most widely and easily accessible to farmers, the option is this genetically engineered crops. So there are institutes in India and abroad who are actually studying how to genetically modify various varieties of crops and ultimately improve their properties. So what are properties? So before going into what are properties of crops are improved, let us understand what do you mean by genetically modified organisms, or who are called genetically modified organisms, or GMO. So plants, animals, bacteria, or fungi who has been, whose genome has been altered by addition or deletion of particular kind of gene. Okay. So these organisms are called genetically modified organisms. Okay. So we are altering the genes, hence the name genetic modification. Now in crops, people use genetic modification or genetic engineering to improve properties or add properties like they should be tolerant to abiotic stresses like drought, famine, okay, then more salt content in the soil. So these kind of abiotic stresses means non-biological stresses, which often in normal condition affects the yield of the crops. But if we can introduce some kind of gene in the crops, which makes it tolerant to these kind of abiotic stresses, then even under drastic conditions like drought, people will have food to eat, correct? Then crops are also made, less dependent on chemical pesticides, okay, by improving its properties to fight the pesticides instead. Then people often suffer, the farmers often suffer post-harvest losses, which is actually a significant problem when you're talking about tons of crops being lost, right? So to overcome this problem, genetic modification are done to the crops so that their shelf life increases. Another reason why crops are genetically modified is to increase the nutritional value of the food. So people have actually developed a variety of rice called golden rice. Okay. And these rice kernels, they are yellow in color because this rice is highly rich in vitamin A. So in countries and developing countries, children who are suffering deficiency in vitamin A, one bowl of golden rice would be enough for them, okay, and be more healthy. Now, a few examples on how biotechnology has been applied for improving crop varieties. First example is BT crops, or the crops who are expressing the gene for BT toxin. So this BT toxin gene is actually coming or is isolated from a bacterium called Bacillus thuringiensis, or is also called B. thuringiensis. So it is a bacterium. What does BT toxin do? So this Bacillus thuringiensis produces this BT toxin, and when this BT toxin is ingested by insect, it basically kills them. Okay. So different kind of BT toxins are there to kill varieties of insects like Lepidopterans. This is a class of insects under which tobacco birdworm, armyworm, etc. These are the examples. Other class could be Coleopterans. So under this beetles come, then dipterans, under this flies and mosquitoes. So basically, these bacteria, Bacillus thuringiensis, by producing the protein Bt toxin, it can kill insects of a variety of. It can basically kill a variety of insects like flies, mosquitoes, okay, beetles, etc. It can kill them, but the prerequisite is the insect has to digest that particular toxin. Then only their action will happen. So how this knowledge is actually helping us to, make better crops. So just understand first how this toxin works, and then we'll see how the gene for this toxin is helping us to create better crops. So this bacteria, Bacillus thuringiensis, it produces the toxin, Bt toxin, but this toxin is produced in its inactive state, which is also called protoxin, which is why uh, this particular toxin protein doesn't affect the bacteria itself. Okay, because in bacteria it is present in inactive state or is present as a protoxin. Now when an insect eats this protoxin, okay, when it eats it, so inside the gut of the insect, the pH is quite alkaline, means greater than 7 pH is there, because of which um, it is the right condition. The toxin gets the right condition to become activated. And when it activates, it will then bind to the cell surface of epithelial cells in the gut, okay, and will cause creation of pores on the cell surface. So because of creation of pores, there will be more and more intake, of water, like due to osmosis, right? And because of which the epithelial cells of the gut will swell up, and ultimately the cells will burst. That is, it will get lysed. So there will be perforation in the gut of the insect, which ultimately causes death of that particular insect who have made the mistake of ingesting this toxin. So as I told that there are varieties of Bt toxins for killing various kinds of insects. So what this means, that Bt toxins are quite insect group specific. So Bacillus thuringiensis is a type of bacteria. So this bacteria has various kind of strains. Each kind of strain will create a variety of Bt toxin, and that variety of Bt toxin will be effective against a particular type of an insect. So people have to first study those. Once they have understood this and mapped out which all Bt toxin are effective against which all kinds of insects, then the gene encoding for the Bt toxin, it is incorporated into the genome of the crop, okay, where you want that particular toxin to secrete and ultimately kill the insects which have dared to attack this plant. So so the people who are studying these crops like cotton, so they found that a certain type of ball worm is very much frequent in infecting cotton crops and ultimately resulting in loss of the harvest. So they have identified a particular type of Bt toxin which is effective against the bollworm that is infecting the cotton. So what they did, they took this gene for this particular type of Bt toxin, and by using genetic engineering techniques, they introduced this gene into the genome of the cotton plant. Now all the plant cells will start producing this Bt toxin, and whenever this insect bollworm infects or tries to attack the cotton plant, it will die. Okay, because it will eat the Bt toxin, and ultimately you know what is the mechanism that will kill the insect. So Bt toxin are actually encoded by a family of genes called XII. So XII is the name of the family of genes which encodes for the Bt toxin protein. For example, under this XII family of gene, there is XII2AB, XII1AC. Now these two genes um, actually secretes Bt toxin that can kill cotton bollworms. The example just I gave. Similarly, there is a gene Cry2AB whose Bt toxin can kill the insect corn borer. So when these genes are, when they say Cry2AB or Cry1AC is incorporated in the cotton plant, then it will, that the plant will be called Bt cotton. Okay, because now the cotton plant will start producing the Bt toxin, which will ultimately will kill the pest cotton bollworm. Similarly, people have incorporated this Bt toxin gene or the gene coding for the Bt toxin in variety types of crops and made cotton, Bt cotton, Bt corn, Bt rice. Okay, so these are new genetically modified crop varieties, etc. Now, next, before moving on to another strategy of making crops foolproof from pest, let us first understand a technique of called RNA interference or RNAi. Okay, what we have to first understand what is RNA interference or RNAi. Now, this particular mechanism actually is a cellular defense mechanism, means cells use it to protect itself. And it is present in all eukaryotic cells. So it is also present in humans as well. This is a process, okay, and that acts as a cellular defense mechanism present in all eukaryotic cells. Now, this process is also called by gene silencing with the name gene silencing or gene knockdown. So why it is called gene silencing or gene knockdown? You will understand once we understand how this process works. Now, what happens in this method? Here, a particular type of mRNA is stopped from getting translated. Means it is stopped. Getting translated means it won't make protein. So that is called silencing. Okay. So it is made silenced. Okay. Like chup ho jao. You cannot get translated. How it is done? It is done by complementary binding of a piece of RNA with this particular type of mRNA. So that results in formation of a double-stranded RNA. Okay. Now there are certain kinds of proteins recognizes the formation of this double-stranded RNA, where one strand is the mRNA and another strand is a small specific type of RNA. So this enzyme one enzyme will come. It will then cleave out the mRNA and will not allow its expression anymore or won't allow its translation. So you may not have understood, it's fine, but now look at this figure. So here what is happening? This is an example of how RNAi or RNA interference is taking place in a eukaryotic cell. Now suppose uh, a retrovirus, this is the retrovirus, okay, it has a genome, RNA genome, right? And within this retrovirus, this genome is double-stranded. Means retrovirus has a double-stranded RNA as its genome. Now, when this retrovirus comes and infects animal cell, like say human cell, then what happens? Let us see. So this is the virus. It has sit on top of the it have very nicely gripped itself to the surface of the human cell and released its double-stranded RNA. Now this double-stranded RNA is in cytoplasm, okay? And it first gets recognized by an enzyme called dicer. So what this enzyme do? This enzyme, as the name suggests, dice out. You must have heard dicing the potatoes, like making into small pieces. Similarly, this dicer enzyme cuts this double-stranded RNA into small pieces. Now, this is how the pieces have been formed. Okay, in this step. Now, each of this piece is called. Now, it will be called siRNA. Also, siRNA stands for small interfering RNA. Okay, so siRNA is formed after dicing by the enzyme dicer of the double-stranded RNA that has come from this particular retrovirus. Then another uh, enzyme complex, means it is a very big enzyme complex consisting of varieties of enzyme called risk complex. Okay, it comes and uh, binds to one of the siRNA. So say it risk complex comes and binds to the siRNA that is highlighted within the green box. Okay, so once it binds, once the risk complex binds, what it does, it unwinds this particular double-stranded RNA. So this is still a double-stranded RNA, but uh, the cleavage has been made in such a way that we get small pieces of double-stranded RNA. Now, each small piece of double-stranded RNA is called siRNA. So this complex comes and unwinds one siRNA molecule, exposing two single-stranded RNAs. So one will go from 5 prime to 3 prime, another will go from 3 prime to 5 prime. Now, if you recall your understanding from the transcription and replication processes, so this strand that is running from 5 prime to 3 prime is called the sense strand, okay? And the strand that is running from 3 prime to 5 prime is called the template strand, or also called the antisense strand. Now, these two single-stranded RNAs are there. Now, what happens? Now, suppose another, uh, suppose another retrovirus has come and before this particular virus and have already escaped or somehow escaped the dicer and the risk complex, because of which its double-stranded RNA has gotten integrated with the genome of the human cell, okay? And because it got integrated, the protein, the mRNA corresponding to the viral genome got transcribed. So this green one is that unfortunate, or you can say the smart uh, viral mRNA that somehow got expressed, and it escaped this dicer and risk complex. So if it got transcribed, the next step would be that it will be translated within the cell. So if it gets translated, it will make viral proteins. And now these viral proteins, if gets assembled, it can create many more new progeny of this retrovirus. So that would mean that the human cell that we're talking about has been successfully hijacked by the retrovirus, and it will not be able to do its normal functioning. Now, if something like this is like happened or going to happen, because of which many viral mRNAs are floating around the cytoplasm of the human cell. Okay, then how these viral mRNAs can be stopped? So here at this step, two single-stranded RNAs are there, right? That the risk complex and the dicers have been able to make. So this complex now what will do? It will come and you know uh, digest or destroy this 5 prime to 3 prime strand. It will destroy it because it is not going to do the job of RNA interference. This 5 prime to 3 prime segment of siRNA, it cannot form complementary binding with this viral RNA because the virus RNA is running from 5 prime to 3 prime, so it cannot. Uh, I hope this makes sense. Whereas the antisense strand that is running from 3 prime to 5 prime, it will be left as it is by the RISC complex because this antisense strand will now contain the base sequence which will be complementary to maybe a small portion of already existing viral mRNA. So this antisense strand will go around in the cytoplasm and hunt for this viral mRNA. So once it successfully finds this viral mRNA, it will go and bind to the region to which it is complementary. So because of which this kind of double-stranded mRNA structure is now formed. Double-stranded mRNA structure is now formed. So once it's formed, the RISC complex is again alerted. Okay. So since it is a complex of enzyme, it releases a group of enzyme that is called slicer enzyme, which are pink in color. So this slicer enzyme goes and binds to this double-stranded RNA consisting of one viral mRNA and antisense RNA from the siRNA molecule. Okay, so once the slicer molecules or slicer enzyme binds, it will fragment out the this double-stranded RNA molecule. It will make it into fragments, because of which the viral mRNA can no longer undergo translation and can no longer produce the viral proteins. Okay, so basically the viral mRNA, uh, like how many molecules there may be, they are all destroyed. So here in this step, where there are several molecules of siRNA that are formed here all of these siRNA can go and they can be unwinded and separated by RISC complex, and all of them can go and hunt for all the viral mRNAs present in the cell and stop them from getting translated. This is the mechanism of RNA interference. The RISC complex actually stands for RNA-induced silencing complex. Okay, so this is the full form of RISC. So this is how RNA interference mechanism works. And I've taken the example of how retroviruses are stopped by this RNA interference mechanism from expressing the important viral proteins and then stopped from making more and more retroviral progenies. So how this is important to us in creating, crop varieties which are itself resistant to pest. Let us understand. So there is a nematode called Meloidigine incognitia or M. incognitia. This nematode is known to infect the roots of tobacco plants, okay, because of which the tobacco plant is not able to produce sufficient yield, means its yield is reduced. So the farmers are very worried about it, right? So we have to do something about it. So once way we have to, what we have to do is stop this particular nematode from infecting the tobacco plant. So they have achieved it by using the RNA interference mechanism, which we just studied. So how it works in tobacco plants. So the tobacco plants are genetically modified. How? Uh, Scientists have recognized that few genes are there which are very important for the survival of this nematode M. incognitia. So they took one of those essential genes and using an Agrobacterium vector, they introduced it into the tobacco plant cells and ultimately created genetically modified tobacco plants. Okay. Now, um, the introduction of this gene is such that it will create double-stranded RNAs, what means this gene when transcribed within the tobacco plant cell, it will create double-stranded RNA molecules, okay, where there is both the antisense and the sense strand. So this kind of double-stranded RNA molecule will be detected by the RNA interference mechanism, okay. It will be detected by all the enzymes involved in this RNA interference mechanism. So there comes the dicer enzyme that will cleave the backbone of this double-stranded RNA and create small pieces, which are called the siRNA or small interfering RNA. Now, this siRNA will be then, each of the siRNAs will be then unwinded and separated by the enzyme of risk complex, okay, risk or RNA-induced silencing complex. So because of which ultimately what you will have only the antisense strand available inside the plant cell because the sense strand will be destroyed by the risk only. Okay. So when this nematode dares to infect this genetically modified tobacco plant, what it will do, in order to survive in the plant, it will try to transcribe this essential gene highlighted in purple and make the mRNA 5 prime to 3 prime in the plant cell, okay, because this nematode needs to survive inside the plant cell, so it will produce this mRNA in the plant cell. Now this genetically modified tobacco plant have this uh, antisense strand, antisense RNA strand, which is actually complementary. As you can see, it is complementary to a small section of mRNA, this 5 prime to 3 prime mRNA, which is coding for a important protein. Okay. So because of this binding, the risk again is activated and it will send in the slicer enzymes to cleave this mRNA of M. incognitia in the plant cell and will ultimately won't allow uh, nematode to survive in the plant cell, because of which the tobacco plants will remain healthy. So this is how RNA interference mechanism is used to protect the tobacco plants from its parasite uh, M. incognitia, or M stands for Meloidigine incognitia. Okay, so this is all about how biotechnology is used in agriculture, means how biotechnology is used in agriculture, where we have only seen the examples of how crops are improved, genetically improved to make them pest resistance. One is the example of Bt crops. Another is the example of genetically modified tobacco plants, which are protected from the nematode M. incognitia by utilizing its RNA interference mechanism. Now, in the area of medicine, that is in healthcare, uh, biotechnology again has played a very important role. Whatever proteins which are made through recombinant DNA technology, that is, which are collectively called recombinant therapeutics, they are actually much more safe and effective because you are ultimately going to use a gene taken from human only and then introduce into a bacterium, and ultimately you get a protein which is of human origin. Okay, but the proteins, traditional proteins that are used before the arrival of recombinant technology, they were all taken from non-human sources like animal sources. So because of which, when those proteins were injected into the bloodstream of humans, they induced unwanted immunological response. That is why I said that recombinant therapeutics like proteins, etc., they are more safe and effective than these proteins taken from non-human sources. Okay, now let us see some of the important applications of biotechnology or the techniques of biotechnology in medicine. First is the making of genetically engineered insulin. Let's talk about that. How people actually made genetically engineered insulin molecules or insulin hormone. So if you have grandparents in your home, or if any of your parents who are suffering from diabetes, you might know that their blood the insulin concentration is quite less, because of which the blood glucose levels are high. So in extreme conditions, when the situation cannot be managed by medicine alone, doctors recommend them to take insulin hormone, okay, intravenously, like directly injected into the vein, so that this insulin in the blood can manage their blood glucose level. Okay, because at those critical times, their pancreas can't secrete the required amount of insulin. That is why exogenously you are providing insulin into the body. Now, often, like before biotechnology came, what people used to do is that they used to take insulin from the pancreas of slaughtered cattle and pig, because cattle and pig are also mammals like us. So they thought their insulin be working, like will work on us as well to control the blood glucose level. But since they are of different species, some species specific properties will be there, which is going to be alerting the immune response of humans. That is why the patients who have taken this insulin of pig or cattle, they have displayed immune response, like several types of immune response, which might be some kind of allergy or maybe an extreme thing. Okay, so in order to prevent this critical situation, people in 1983, few people in 1983, they decided to use the recombinant DNA technology to make recombinant insulin. Okay, so it was first brought into market by the company Eli Lilly. So what happened is, uh, in humans and or other mammals, in pancreas, insulin normally is produced in inactive state. Okay, but after removal of a particular peptide, one peptide component, that is C peptide, insulin immediately transforms into its mature and active state. So here in this figure, I have shown uh, the how the native inactive insulin structure looks like. Okay, so this is your proinsulin. So there are three chains, peptide chains, A, B, and C. And A and B peptide, they are linked by another peptide called C peptide. Okay. Now, after the removal of this C peptide, this A peptide and B peptide, they are linked together by disulfide bond. So here you can see the disulfide bond is there. Now, when this kind of structure is there, this is the structure of mature insulin, or the insulin hormone, which is in active state, which is now going to do its job of regulating the blood glucose level in the blood. Now, this is how the structure of proinsulin and mature insulin differ from each other. So what the people in Eli Lilly company did, they made recombinant insulin. How did they do that? They took plasmid A, and within the plasmid A, they inserted the sequence for A peptide. And they took another plasmid, say plasmid B, there they inserted the sequence for B peptide. And these plasmids were transformed into E. coli cells. So, as you know, we can, this is called gene cloning, and then they ultimately expressed this peptide A and B in two groups of E. coli cells, and got pure A peptide and pure B peptide separately. And when these two peptides were mixed together and allowed to form disulfide bonds, what they got recombinant insulin. Okay, they got the insulin in the uh, in its mature state, in its active state, which can now be injected into the bloodstream, and it will do its job of regulating blood glucose level. So this is how recombinant insulin is made. Now, another area where biotechnology has really helped in making people's life better is, development of gene therapy. So, what is gene therapy? Basically, it is actually a collection of methods that allows correction of a gene defect that has been diagnosed in a child or even in embryo. So, we can use gene therapy techniques to correct a gene defect, okay, which luckily got diagnosed in a small child, or even when inside the mother's womb, that is an embryo condition. We can correct that gene defect. So what people do, they first identify which gene is in defective state. Then they insert a normal copy of that gene into the person's cells or tissue. So once they do that, this normal copy of gene will get expressed, okay, inside the target cell or tissue, and will secrete the protein, which will then ultimately help the person elevate its disease symptoms. So for one such disease, genetic disorder, where gene therapy has been applied, is called adenosine deaminase deficiency. So this is a genetic disorder that children have from birth, where in these individuals, this enzyme adenosine deaminase is not produced because the gene for this enzyme is deleted, and it is happening, at the gene level, while the fetus is in the womb. So because of this, because of deficiency of this enzyme adenosine deaminase, the immune system of the individual fails to function normally. Okay, and that person becomes very much susceptible to even simple diseases like cold. So the life expectancy of that individual decreases. So various treatments have been developed, various kinds of treatments have been developed for correcting this disorder, not correcting, but for making the life quality of the person having this deficiency, ADA deficiency, So people have tried bone marrow transplantation, then gene replacement therapy, but all these therapeutic techniques, they are not a complete cure. Okay, they just temporarily elevate the symptoms of ADA deficiency and require repeated procedures. So people then try to develop a better technique using gene therapy principles. Okay, so what people did from ADA patient, okay, they isolated their that person's lymphocytes from the blood. They took a blood sample, and from the blood sample, they isolated the lymphocytes. Now, and within this lymphocytes, they introduced a functional, that is a normal ADA gene, okay, a functional copy of normal ADA gene using vectors like retroviral vectors. Okay, so now because of this, these genetically modified lymphocytes will start expressing the uh, adenosine deaminase enzyme. Once these lymphocytes are, successfully uh, transformed, and then they successfully express this enzyme, these genetically modified lymphocytes are returned back into the patient's body. So, and so when these lymphocytes are circulating inside in the blood veins and blood vessels of the patient, they will secrete the enzyme and rectify the condition or improve its symptoms. But since lymphocytes are not immortal cells, they have a limited lifespan of few days. So this transfusion means this infusion of genetically modified lymphocytes have to be repeated again and again, so that the patient is not made completely devoid of adenosine deaminase enzyme. But if somehow um, doctors are able to insert a functional copy of ADA gene in the fetus, okay, or in the embryo inside the mother's womb, that is at a very early stage of development, then this disease can be cured permanently. Okay. So for another way how the biotechnology has really helped the medicine field is the introduction of various kinds of molecular diagnostic techniques. Means people have developed diagnostics techniques which can find out or detect any gene defect or at the RNA or a DNA level. So this kind of work is made possible through the biotechnology techniques or technology, okay, through this field, biotechnology. So traditionally, any disease is diagnosed by, doctors asked for patients to go to uh, give their urine sample or blood sample, and then they analyze the contents of their urine or analyze the contents of the serum. So that is how they detect if any kind of foreign pathogen or foreign proteins are present in the blood or urine. But by the time a particular pathogen like bacteria or virus is able to secrete large amounts of foreign proteins in the blood and urine, it means that its number or its concentration has increased a lot in the body. Okay. So this kind of detection is only is not able to give an early detection, means at a stage when the concentration of foreign antigen in urine and blood is not that much. Means the symptoms of the person is like the person is not showing any symptom for a disease. So usually when symptoms come to a person, then only they go to doctor, and then doctor refers this test. But by that time, if the disease is a fatal one, not much can be done. But due to the use of molecular diagnostic techniques like PCR, polymerase chain reaction, then using recombinant DNA technology, then using an ELISA, like these are only few examples. Today, there are much more which you will study in your higher classes. But these kind of molecular diagnostic techniques actually allow doctors to detect a particular kind of a disease at a stage when its symptoms has not yet started to show. Okay, so here ELISA, you must be hearing this term for the first time. It actually stands for enzyme linked immunosorbent assay. We will not go that much in detail, but you should know what ELISA stands for. So you must have heard during COVID-19 that people asked to do RT-PCR test. Okay, so that RT-PCR test is nothing but the PCR mechanism that we studied in this chapter. Okay, means in this unit of biotechnology. Okay, that is the same thing. Okay, so uh, at that time, people said PCR test will show whether a person will has COVID or not. Similarly, PCR test can also be used for detecting other kinds of diseases as well. Why? Because PCR actually amplifies even a small amount of DNA. Like if you given even a one molecule of DNA, after 30 cycles, you will get billions of copies. So billions of copies of a particular DNA fragment, right? And when you run through agarose galactophoresis, you will get a much more thicker band, and you'll be able to say with more confidence that this particular disease has happened or not. So that is why PCR is a very, very powerful tool for disease diagnosis. So um, So apart from the COVID-19, earlier than that, PCR has been used for detecting diseases like various kind of diseases actually, like rheumatoid arthritis, cancers, etc. And it can also help doctors to detect if a person is carrying any mutated gene, which can ultimately cause a particular type of cancer. So in those individuals, the treatment can be started early, or uh, and so that the person doesn't reaches to an extremely fatal stage of a cancer. Whereas ELISA, it stands for enzyme linked immunosorbent assay. It is actually based on antigen antibody reaction. Okay, how a particular antigen binds to a particular type of antibody, and then due to this binding, you're able to say um, this foreign antigen is present in your blood or a urine sample. So we will not go that in that much detail. So for today, we will stop our discussion, okay, on this chapter. The next class will cover up very small topics and ultimately finish this unit of biotechnology. Thank you.